My name is Barney Geddes. I'm an assistant professor here in the Department of Microbiological Sciences at North Dakota State University. Farmers are likely familiar with the root nodules they can see on soybean roots after they've inoculated their seed. These nodules are formed by bacteria called rhizobium that can take nitrogen directly out of the air and provide that as a form of fertilizer directly to the plant. Now this is super important, of course, because first of all, fertilizer is incredibly expensive. The more we can get directly from the air, from this natural relationship, the better. And second of all, it's really an environmentally sustainable process. So when you start thinking about sustainable farming with soybeans, making sure we're maximizing that natural relationship that they have with rhizobia is incredibly important. So what we're doing here is we're loading a nanopore sequencer, which allows us to read the genomes of these rhizobium bacteria. One of the things we've been doing is developing molecular tools that can allow us, basically like a soil test, to really quickly understand the populations of these rhizobia as they're existing in farmers' field soils. So here we're just culturing these rhizobium bacteria that we've isolated from root nodules from North Dakota farms. And so after we've isolated them, this is the kind of thing we might take and go test in the greenhouse to see the quality of those rhizobia. So that has a couple important impacts. One of them is that we're hopeful that at some point we might be able to provide recommendations regarding inoculant to farmers. But perhaps even more importantly, it's just giving us a really accelerated ability to do agronomy, to understand different factors that are impacting rhizobia populations in the soil. Scenarios where a farmer might want to think about getting more rhizobium back into their soil by inoculating again. In the spring when they're planting their beans, maybe they've had a flood or a drought, and we're thinking we can get to a much deeper understanding of how these different factors impact nodulation success and whether it's profitable to actually inoculate the soybeans. So when I think about that research in the longer term, where is it going? My vision for that is really to have this ability to give a farmer a deep understanding of their soils and integrate this information about the biology, the rhizobia that's happening there, with understandings of soil chemistry and everything else. And I think we could get to the point where we might even be able to predict, which field is going to harness this symbiotic nitrogen fixation relationship the best. The other stuff that we're interested in is looking a little bit even further into the future, starting to think about what opportunities are there for us to boost the nitrogen fixation that's actually happening through rhizobia in soybean fields. And a lot of that lies in thinking about genetics of the plant, thinking about genetics of the rhizobia that either exist there naturally in the soils or that we might want to apply as inoculants. And so there's a few things to focus on here. One is that are there opportunities within breeding to be ensuring that as the public breeders such as those here at NDSU are thinking about elite lines, we're thinking about lines that can maximize that nitrogen fixation process. So we've got a great collaboration with Carrie Miranda in the Department of Plant Sciences where we're thinking about that specifically in the context of drought, because drought can be really hard on this symbiotic nitrogen fixation, and we're hopeful that there could be scope to develop lines in the long run that do a lot better in terms of sustaining this symbiosis when they're under the slightly more challenging conditions of these sort of drought-like conditions that we seem to be experiencing more and more lately. So one thing that started to emerge from our research is that not all rhizobia are created equally, right? So although you see nodules, and those maybe came from an inoculant you might have applied this year or even a few years ago, they also might not have. They can come from these natural rhizobia that exist in the soil. And not all of those natural rhizobia are as elite as you might want or are able to perform their job as effectively as we'd like them to be able to. And so we are thinking a little bit about now in the longer term, should we be taking a more tailored approach to inoculation? Should we be finding specific rhizobia that are really really thrive in western North Dakota soils, for example, or that really have a perfect match with the cultivars we might release from our breeding programs here. So I'm hopeful that we could get to the point where nitrogen supplementation doesn't do anything, where you want to get every little bit of nitrogen the plant needs coming from that symbiosis, and hopefully even leaving a little bit behind for the next crop.